Hello, welcome back. Today we're going to look at an important topic in biochemistry, which is one of the most fundamental aspects of experimentation in biochemistry, or even as a matter of fact, any branch of life sciences, or perhaps which cuts across different disciplines of science, is uh, buffers. Okay. Uh, most of you who might be undergraduate students or postgraduate students or even involved in research in various capacities might know about buffers or might have worked with buffers. So this would be a fairly easy topic for me to cover, but it's still important to know why is pKa of a given acid or pKb of a given base, which is chosen for making buffer, is one of the most important considerations that one has to keep in mind when preparing buffer. Okay, by definition, buffers are aqueous solutions which are designed to resist changes in pH when small amounts of acid or bases are added into it, such that the pH of the experimental medium is not subject to variations and remains at the pH that you want to work with. Think about a situation where you're trying to homogenize cells, and in case you end up lysing lysosomes, then without a buffering component, the lysosomal enzymes may degrade critical components of a homogeneous system, because lysosomal enzymes are all degrading enzymes, okay, the enzymes that bring about degradation. But if you maintain the pH of a solution in which you are homogenizing the cells at cellular pH, cytosolic cellular pH, then at that pH, lysosomal enzymes will not be active. And because they are not active at cellular cytosolic pH, your cellular components are, they remain unaffected by lysosomal enzymes, because lysosomal enzymes are acid enzymes. Okay, they're active at lower pH. So that kind of very quickly underlines the importance of buffers. Buffers are aqueous solutions, as I said earlier, which are meant or designed for resisting changes in pH when small amounts of acid or bases are added. And buffers are made of a weak acid and its conjugate base, or a weak base and its conjugate acid. It's as simple as that. Okay, you can look at the Henderson-Hasselbalch equation, determine the concentration of the weak acid in the conjugate base, or the weak base in the conjugate acid that is required to prepare certain volume of buffer to be held at a certain pH. Okay, it's as simple as that. I've given a couple of examples here. This is a combination which involves sodium acetate as the conjugate base, along with the acid, which is acetic acid. This is sodium dihydrogen phosphate, which acts as the acidic component of the buffer, and disodium hydrogen phosphate, which acts as the basic, or rather the conjugate base of the buffer. You can replace sodium with potassium as well. So all the varieties are available as commercial compounds. You can use whatever you like. But what is most important is, uh, most often what happens is, when someone is out to prepare a buffer, they would simply look at the protocol and just prepare the buffer. But it's always nice to appreciate the importance of pKa, because in case if you find yourself in a hypothetical situation where you have to set your the pH of your buffering solution at certain value, for which no ready-made recipe is available in any literature. I mean, it's not likely to happen. I'm just talking about a hypothetical situation. Let's say that you have to set the buffer at a certain pH, and you're not able to come across a ready recipe which tells you what to put in the buffer. Then invariably you will have to resort to finding an acid or a base. They have to be weak. Okay, I mean, they shouldn't be something that completely ionizes in water, whose pKa or pKb respectively is closest to the pH you want, preferably within 0.5 units, but even 1 unit is fine. Okay, so this is the background with which I'm making the idea. I think there are a lot of videos online about how to make buffers, or that explain buffering mechanism, but I think it's still important to appreciate the pK of a given acid, or pKb of a given base, when it comes to making the right choice of an acid or a base respectively, for making either an acidic buffer or a basic buffer. Okay, before we get into pKa, just let's look at the dissociation constant. Okay, Now, what we have here is a very simplistic depiction of an acid ionizing, okay? And I would like you to appreciate that there is a reversible sign. That means uh, this is a weak acid which does not go complete ionization. That means if you take a million molecules of acid, the H of the acid here represents the proton that eventually will dissociate, and what will remain as the anion will be the conjugate base. It's as simple as that. If you take a million molecules of the acid, not all the million molecules will ionize, depending on the strength of the acid, which is given by the term dissociation constant. A few of them will ionize, which will enter into equilibrium with the unionized acid. Okay, that's as simple as that. So if you're looking at a weak acid which is ionizing to give you protons and the conjugate base, the dissociation constant would be simply the ratio of the product of the ionized protons and the conjugate base divided by the concentration of unionized acid. Okay, so it's simply a concentration of protons into concentration of acids divided by the concentration of unionized acid. And uh, higher the Ka of an acid, stronger will be the extent of dissociation of the acid, simply implying that it will be relatively a stronger acid compared to something which has got relatively lower Ka dissociation constant. Okay, it's as simple as that. Now, the problem with the term Ka itself is it's a very small number for most weak acids. Like, you look at acetic acid, okay? The Ka is 1.7 into 10 raised to 5, sorry, minus 5 molar. It's a very small number, right? If you were to work with this in your calculation, this poses a lot of problem because the number is very small. There are a lot of zeros after the digit, right? So it's going to be difficult. So what you do is you introduce a term called as pKa. Okay, it's similar to pH or pOH or whatever, right? P function for any given parameter is simply negative log to the base 10 of that parameter. It's as simple as that. Okay, so pKa is nothing but negative log to the base 10 of dissociation constant. And I'd like you to appreciate, you don't have to memorize anything, it's just for your understanding. The dissociation constant of acetic acid is 1.7 into 10 raised to minus 5 molar. And the pK, if you calculate, if you take negative log of that value, it will be somewhere close to 4.7. Okay. Likewise, H2PO4 minus, this is the acidic form that is left behind after the first ionization of the phosphoric acid. The reason why I've taken this and not the first one is because this is most relevant in terms of the buffering capacity of the cytosolic components. Okay, I'm not going to go into that. I just mentioned that if you're looking at human body as such, we are all blessed to have buffering components inside our body. In what typically is described as the extracellular fluid, the buffering component is bicarbonate buffer system, and in intracellularly it's the phosphate, particularly this phosphate and its conjugate base that operates as the buffering component, because its pK is 7, close to 7.2, because its dissociation constant is 6.2 into 10 raised to minus 8 molar. Okay, that's something you got to appreciate. Now we're getting to a point where we're starting to appreciate the importance of pKa, because if you want to prepare a buffer at a certain pH, then you got to make the choice in terms of using the right acid or the right base, such that the pKa or the pKb respectively is closest possible to the pH that you desire. If it is exactly equal, all the more better, because the buffering capacity of such buffers, when the buffer is held at the pKa of the acid or the pKb of the base that is used, the buffering capacity of such buffers is going to be the best for that given concentration of the buffer. Okay, so I wanted to appreciate the value of pKb or pKa when it comes to preparing buffers, and to look at that, we really need to go to the titration curve of. I'm going to take a simple example. What we're looking at here is a titration curve of acetic acid, and there may be some inaccuracies. It's just a hand-drawn diagram, but I'm sure you'll appreciate it. If you take acetic acid in a beaker, okay, and just take a little bit of acetic acid, dissolve it in a little bit of water to get a certain desirable concentration, and measure the pH, okay, right? It would be pretty low because it's an acid, right? It would be pretty low. And as you add small increments of sodium hydroxide and measure the pH, initially there will be fairly strong increase in the pH because you're adding a base into the solution. there will be an increase, and you will see a point eventually along the titration curve where the pH does not change much for a while, despite adding incremental amounts of sodium hydroxide. And this is the most important thing I'd like you to appreciate. From the graph itself, it is quite visible that the weak acids or the weak bases have an inherent ability to resist change in pH around their pKa. Okay, now if you were to do this experiment, you'll get a typical graph like this for monobasic acids, which will be this shaped. Okay, you will see definitely a flat region where the pH won't change much despite adding incremental amounts of sodium hydroxide, and the midpoint of that line taken back to the y-axis, which is the scale for pH, will tell you what the pKa is. Okay, this is experimentally what you determine, and antilog of that would be dissociation constant as well. Okay, so it's as simple as that. But anyways, right? One. Okay, so you got to take analog function eventually to be able to determine the Ka, but this is the pK, and right now you can appreciate the fact that you took just acetic acid, and as you added small amounts of sodium hydroxide, the pH increased. But as the pH is increasing, certain amount of acetic acid is being neutralized by sodium hydroxide. So what you're getting is sodium acetate. That is the conjugate base, right? When you hit the right concentration of conjugate base, you will also see that the buffering capacity kicks in, because you would have attained the right concentration of acid and the base as described by the Henderson-Hasselbalch equation. And in this region, the buffering component really exhibits one of the most resilient features, which is the fundamental basis of biochemistry, which is buffering capacity, right? So from here to here, even if you add a base, the pH is not going to change, right? Now the midpoint of this flat line taken back to y-axis is your pKa. The value of this curve in terms of helping you understand the importance of pKa is that if you hold your buffering buffer at this pH, okay. Now I think this was some what was the pKa of acetate? 4.67, right? So if you prepare an acetic acid buffer which is set at 4.76 pH, then for as much as this distance on the right side and as much as this distance on the left side, it won't change, it won't demonstrate significant change in the pH when you add base on this side or when you add small amounts of acid on this side. It shows that resilience, which is what typically the inherent feature of buffers is, right? So that is what is the importance of pKa. So next time when you prepare buffer, please appreciate the fact that whatever pH you're preparing the buffer for, the pKa or the pKb of the acid or the base will be close as possible to the pH that you desire. You can't take acetic acid and expect you to get a good buffer which works well at pH 8, because beyond this point, beyond the pH of 5, right, or whatever, right, 5.5, small amounts of acid or when you add small amounts of base, there will be drastic change in the pH. It won't resist the change in pH. And below this point, whatever it is, right? If you add small amounts of acid, then the pH will change drastically, and that's the value of pKa. So next time when you prepare a buffer, please do appreciate the fact that you're preparing a buffer with an acid whose pKa is closest to the pH that you're trying to set the buffer for, right? So this is the value of pKa when it comes to preparing a buffer in terms of having the right pKa and choosing the right acid that has the right pKa for achieving the desired amounts of concentrations of acid. And obviously, its conjugate base would be your the other part of the buffer, right? So I hope you understood this, because this concept, comprehension of this concept is very, very important to be able to do well in research, because if you understand one concept well, it's likely that you'll understand other concept well as well. Okay. Now, other thing I would like you to appreciate is at this pH, which is the pKa of the acid, right? The concentration of acetic acid will be left behind as acid will be exactly equal to the concentration of ionized acetic acid. So in other terms, pKa is simply the pH at which pH at which 50% of the acid is ionized. It's as simple as it. So next time, if anyone asks you what is the simplest description of pKa for an acid, just tell them that pKa is the acid, pH of the acid at which you will see 50% ionization of the acid. It's as simple as that, right? I hope you'll appreciate this fact. Now, what we'll do is we'll move on and look at the pKa and Ka values for phosphate buffer that you generally prepare in the lab. So for H2PO4 minus, this is the acidic component, and HPO4 minus 2 would be the conjugate base. Okay, for this system, the Ka is 6.2 into 10 raised to minus 8 molar, and the the Ka, right, and the pK will be 7.2, and this is what is closest possible you can get to cytosolic pH. And that's why whenever you desire a buffer which is close to cytosolic pH, when you're trying to do an enzyme assay or whatever, right, which are cytosolic enzymes, will invariably use phosphate buffer, right, most often. The choice of phosphate buffer as the most widely used buffering component in a biochemistry lab is not by accident. It's because of its pK, and I hope you appreciate that fact. It's very, very important. Okay. Now, let's just spend some more time quickly trying to understand how buffers actually work. In order to prepare a buffer, you just need to take the conjugate base and the acid of that of a buffering system as per Henderson-Hasselbalch equation, right? pH is equal to pKa plus log to the base of salt by acid. It's as simple as that. Uh, uh, the derivation of this and the practical problem on utilizing Henderson-Hasselbalch equation to determine these concentrations, I'll do it in another video. But I wanted to appreciate one thing. Okay, now let's just say that you have a beaker. You have a couple of beakers, beaker A and beaker B. Okay, in beaker A, you have 0.1 molar HCl. Okay, it's just a hypothetical situation, right? 0.1 molar HCl. Let's say 20 ml of that. In beaker B, you have 20 ml of, say, 100 millimolar phosphate buffer, whose pH, phosphate buffer, whose pH is, let's say, 7.2, whatever, right? 7.2. 7.2. Now, both are 20 ml. Okay. Now I'm going to add 20 more ml of water into each of these beakers. Okay. The final volume is 40 ml for both. What do you think will happen to the pH of this solution, which originally was 0.1 molar HCl, and what will happen to the pH of this solution, which was originally 7.2 of phosphate buffer, when the volumes are doubled by addition of water? Uh, uh, if you understand this, I think you'll understand the true importance of Henderson-Hasselbalch equation. When you dilute an acid, the pH is going to go up. Okay, but when you moderately dilute a buffer, the pH won't change, because simply look at the Henderson-Hasselbalch equation. You already set the pH at 7.2, so this is going to be constant for this buffer. Okay, and the pK for any acid is constant. It's not going to change for a given temperature. So what really the pH of the solution depends on is the log of the ratio of the salt and acid. And when you dilute something by a certain extent, both salt and acid are going to get equally diluted, so the ratio never changes. It's more like 1/2 is same as 2/4. Okay, so even if you dilute it, the ratio is going to be same, because it's going to be still 0.5, right? So therefore, the pH of a buffer doesn't change when it's moderately diluted, and that's one of the reasons why in any typical lab, the pH, the buffers are prepared at higher concentration and kept in a corner and diluted whenever they want to use it, rather than having to prepare again and again. Okay, it's just a logistical nightmare, because buffers are so frequently used that one wouldn't want to prepare it again and again. They simply prepare a certain amount of buffer at a higher concentration, and whenever they want, they'll draw a small liquid from the reservoir of the stock buffer and dilute it to get the desired concentration, while the pH doesn't change much, assuming that there is no contamination. Okay, so that's the value of Henderson-Hasselbalch equation in terms of helping you understand why doesn't the buffer pH change when it's moderately diluted, right? Very important. Now, quickly, within a couple of minutes, we'll try to also look at how buffers work. I've taken the example of acetic acid, sodium acetate buffer. Okay, let's not worry about what the pH is. It has to be 1 unit on either side of 4.76. Can't be anywhere else. But let's say that you've prepared an acetate buffer, and let's say how it works when you add a small amounts of acid or base. Okay, now when you add a small amount of acid, which is depicted in terms of H plus and Cl minus ions, because you can't simply add the H plus without bringing in the counterpart of the anion. Okay, so let's say that you're adding a small amounts of HCl into the buffering component. The buffering component has sodium acetate. Okay, now sodium acetate is a strong electrolyte. It's going to ionize into an acetate ion and a sodium ion. Okay, now this acetate ion is going to react with proton and push back the equilibrium towards formation of the acetic acid here. Okay, it's going to push back the equilibrium towards formation of the acetic acid. And what happens is, another thing I wanted to appreciate is the product of this reaction between the ionized acetate and the protons that you're bringing in is going to be unionized acetic acid, which is not going to change the pH. Okay, only when it ionizes does it reduce the pH. Otherwise, it won't reduce the pH. So therefore, when you add a small amount of acid, the pH is not going to change because the protons coming in from the acid are going to react with the acetate ions to give unionized acetic acid, because you are shifting the equilibrium as suggested by law of mass action. It's as simple as that. Okay, now let's look at what happens when you add a small amount of base. uh, when you add a small amount of base, the one that's going to help you with resisting the change in the pH is the acetic acid, unionized acetic acid. Okay, so when you add a small amount of base, the hydroxyl of ion of the base will actually draw unionized acetic acid to be ionized into acetate ions and protons, right? uh, as much as base you've added, that much of acetic acid will be ionized, and the protons are going to react with water, they're going to react with the hydroxyl ions to give you water. Okay, and it's not going to change the pH. And uh, you want to, these two are going to react with each other. I mean, you anyway will result in formation of sodium acetate. So the protons that are coming about from the ionization induced by the base is going to react with the hydroxyl ion of the base and going to give you water, and therefore the pH is not going to change. And this is really how a typical acidic buffer would work. And the concept is similar for a basic buffer also. Let's say that you're taking ammonium hydroxide and ammonium chloride as the buffering components. The mechanism would be same. One of them will react with the other such that the buffering capacity is maintained and the pH doesn't change. Uh, so that was the idea of making this video. The whole purpose of making this video was to be able to help you appreciate the value of pKa. This is the most important thing. If you can appreciate the value of pKa as one of the most important considerations that determine what is chosen for making a buffer in terms of acid, or value of pKb in terms of what is chosen for making a basic buffer, then I think you really grasp the concept very well. And I hope after having seen this video, your understanding of what buffers are, how do they act, and how do you make buffers is significantly improved. If you like the concept that has been discussed in the video, please subscribe to my channel. It will only encourage me to make more videos and post such that we all can learn from each other. Thank you so much.